So the major goal for both basic and applied biologists is assessing the condition of their study organism. And condition can seem like an intuitive concept. There's no universally accepted definition of what condition is, which can make assessing it difficult. So a recent study proposed that condition is a product of an organism's genetic, somatic, and epigenetic states. Now, the genotype is inherited and is fixed for life. Somatic state is the sum of an organism's available resources, its degree of parasitism, oxidative state, and age. And the epigenetic state results from events in life affecting gene expression. And these are all important, but I'm going to be focusing on somatic state today when I talk about condition. Now, to assess condition, a lot of researchers use sort of low-resolution measures like a visual fat score estimate or some residual of body mass over a skeletal feature. Or you can use terminal sampling, but that may not always be desired or optimal. A lot of researchers have begun incorporating parasite measures into their assessments for condition. And assessing ecto and endoparasites can provide information on condition at both the individual and the population level. Some researchers are using remote transmitters to track an animal's activity levels and even their metabolic rate in the wild. To complement these data, we can now assess aspects of nutritional health, like total protein levels and lipid profiles from small blood samples. Immune function is also another major component of condition. We can now assess aspects of immune function, such as bacteria killing ability, white blood cells, different types of antibodies from a blood sample. Stress also affects condition and can be assessed from hormones like corticosterone or cortisol, in addition to the balance between oxidative stress and antioxidant capacity. And finally, there's evidence that the microbiome, which is the community of microbes that live in and on an organism, can influence things like digestive efficiency and immune function, which can shape development, rates of development, and even sexual selection. All right, now I'm going to talk a little bit about my proposed work for this coming year, examining the mechanisms shaping avian population dynamics in a human-altered landscape. And the focus of this is going to be on the effects of habitat urbanization, as well as the effects of changes in perceived predation risk. Now, we know that predation risk varies across the landscape, and I'm interested whether birds change their investment strategies in current reproduction and in self-maintenance based on this risk of predation. Now, the three main projects are going to be assessing the physiological consequences of nest site location along this urbanization gradient, looking at maternal and investment strategies along this gradient in cowbirds, and manipulating perceived predation risk as well. Now, for the first project, I'm going to be looking at 6 species of shrub nesting birds that all breed in the same habitat. And 3 of these species are declining, and 3 of them are stable. And I'm interested in mechanisms underlying these differences between these species. And to do this, collaborators and I are going to look at blood parasites like West Nile virus, both in the birds and in the mosquito vectors, as well as endoparasites and intestinal parasites in the birds along this urbanization gradient. And to complement the parasite data, we'll be looking at measures of immune function like bacteria killing ability, white blood cell differentials, as well as aspects of nutritional condition like total protein levels and lipid profiles. And to assess stress levels, I'll be looking at corticosterone, which regulates the stress response, as well as oxidative stress levels and total antioxidant capacity, which together can influence cellular efficiency and even longevity. And then finally, I'm going to be looking at egg size and egg mass, as well as the deposition of different hormones into the eggs, to see if females of these different species allocate resources differentially along this urbanization gradient. Now, for the second project, I'm going to be looking at patterns of maternal investment in the brood parasitic female brown-headed cowbird along the same gradient, using the same techniques to see if female cowbirds adjust strategies along this urbanization gradient based on their host's nest site location. And for the third project, I'll be manipulating perceived predation risk by using playback predator calls and putting predator mounts near nests, and then assessing the condition in the adults and the chicks over time in these sites. My hope from this work is that we can better understand how these measures can be used in the field of conservation biology, to better integrate life history theory and applied ecology, and hopefully move us to being more proactive in our management